Now the next thing that we will be studying is about the humoral immunity. We have read about the cellular immunity. Now it's time to give the importance to the humoral immunity. So humoral immunity is nothing but a branch of adaptive immunity. The B lymphocytes produced antibodies, highlighted B lymphocytes, because we have seen in the cellular immunity, in the cellular immunity, who was the key hero? The hero was the T lymphocytes, right? And over here, the key heroes are the B lymphocytes. And the antibodies neutralizes the extracellular pathogens. In the cellular immunity, it was intracellular. So see, the things which I am highlighting now, please focus more on that one day before your university exams. Although I am giving you the summary tables, but please go through all the highlighted parts that I have mentioned in my notes, right? Next we will see about the components. So the components of humoral immunity that we have, first one is the B lymphocyte. What does B lymphocyte do? It recognizes the antigen via the B cell receptors. You have something called as the BCR. You have something called as the BCR, B cell receptors, that recognizes the antigen. Second is the plasma cells. They differentiate between the B cells that secretes the antibodies. Memory cells, they are the long-lived cells and they respond rapidly on the re-exposure. See, this story of re-exposure now will come multiple times when you will read the chapter, of immunity. So you have to be very thorough with it, and very well you should know that what are the examples of re-exposure. First example I have given you is of chickenpox. Second one, vaccines, because I have given you the example of tetanus, that tetanus provides you a protection for overall 5 years, right? So, fourth one is the helper T cells. What it do? It provides the co-stimulation via the cytokines. It provides the co-stimulation via the cytokines like the interleukin 4, interleukin 5 and the interleukin. Now let's have some molecules and let's see their function. First, we have seen the B cell receptor, that are the BCRs. They are the surface IgM and IgD on the naive B cells. Cytokines, they promote the B cells proliferation. Next you have is the MHC class 2. MHC class 2 is basically present and proceed the antigen to the helper T cells, right? CD4 or CD40 or 40 ligand, they stimulate the signals. Between the B cells and the T helper cells. Next, the story of recognition. So you have antigen recognition. They are done through two mechanisms, B cell activation and the T independent antigens. Via you and your siblings, the process of activation is taking place, right? So in the B cell activation, what is there is there is a binding to the BCR. There is a binding to the B cell receptor. See, I'll draw a diagram for you all so that it will be easier for you. You have something called as the B cell receptor, that is the nothing but your BCR, right? So in the BCR, there is some antigen will come and there will be a binding. There will be binding on the MHC class 2 receptor. There will be a process of binding that will take place on the MHC class 2 receptors. Next is the T independent antigen. So they can activate the B cells without the T cells. They do not require the T cells for the activation. They can activate with the help of B cells as well. So they are mostly produced by the IgM and they have a very weak immune or a memory response, right? Now, let's see about the B cell activation pathway, although I have told you with the help of this cute little diagram, but still. First, there is antigen binding. Again, the story of recognition. The BCR recognizes the antigen. See, if I draw, this is your something called as an antigen. Then there is something called as a BCR complex on the MHC2. So whenever this antigen will come, the BCR complex will recognize it, and they are basically displayed on the MHC class 2. They initiate the signals, and there is the B cells activation. They initiate the signal, and then the process of B cell activation is there. Second is the helper T cell interaction. These helper T cells basically recognizes the antigen on the MHC2 receptor. The antigens that are there on the MHC2 receptor, this helper T cells recognize it. Okay, and what they do? They bind to the CD40 ligand. Okay. Next is there is a release of cytokines which is there. At the last, there is release of certain cytokines. Third one is the clonal expression. The activated B cells now undergoes the process of proliferation. What is the process of proliferation? Process of proliferation is nothing but the process of mitosis, right? So the clonal production of B cell is there with identical specificity. There is a production of the B cell that will take place. Fourth one, last one is the process of differentiation. The plasma cells secrete the antibodies and the memory B cells resides in the lymphoid organ. And tell me, where does the T cells reside, right? Because B cells reside in the lymphoid organ. Where does the T cells reside? That you have to tell me. Next that you need to understand is about the immunoglobulins and their functions. It can be asked as a short note. And if it is asked as a short note now, then this table is very, very important for you. Just write this table when it is asked as a short note, right? So I have told you, you have 5 classes. What are the 5 classes you have? You have G, A, M, E and D. So gamete. You have 5 classes of IgG, not IgG, the immunoglobulins. You have 5 classes gamete, right? The first one is the IgM. So IgM is a pentamer in structure, and it is the first antibody in the primary response that activates the complement system. And it is the largest. See, remember, if it is the largest now, then remember M for M, that is M for massive. Okay, M for M. Next you have is the IgG. It crosses the placenta. So G for it goes. Goes where? It goes through the placenta. It crosses the placenta. It is monomer in structure. And has secondary response and follows the process of opsonization, the process of making the bacteria tastier, right? Then there is complement activation. Next you have is the IgA. It is dimer in structure. What is the point you need to remember? I have also mentioned that. These are your notes, because these are the points, because it can be asked as a MCQs. In the MCQs, you have certain keywords which you need to identify. So that's why I have given you a list. Not separately, I have given you the list with them only, okay? So IgA is a dimer in structure and it is present in the secretions like in saliva and tears, and neutralizes the pathogens. The note that you need to take, they are involved in the mucosal immunity, okay? Next you have is the IgE. IgE is monomer in structure. It is monomer in structure and is involved in allergic reaction. Allergic E. So it is IgE that is involved in allergic reaction. Okay. Whenever you read now, just try to make some mnemonics that you are able to recall on the day of the exams, right? Not the mnemonics that you are not able to recall, but you have made the mnemonic just for the sake of it. So please make some mnemonics from yourself as well, Although I am also giving you certain mnemonics, but please try to make some. Right. Next, you have allergic reactions. They are involved. They bind to the mast cells because mast cells are responsible for the allergic reactions. And if I ask you about mast cells, next thing that should come to your mind is histamine. Okay. I'll make sure some of the things will get into your mind for sure, right? So that's why I'm stressing upon these little words very much. IgD is monomer. B cell receptor is there. They initiate the B cell activation. They initiate the process of B cell activation. Next, understand about the mechanism of antibody action. The mechanism of antibody action. First one is the process of neutralization. What it does? It blocks the microbial toxins from binding the host cells. See, this is the host cells. If the microbes are coming, so the process of neutralization blocks them from coming to the host cells, right? Next is the process of opsonization. I have already told you opsonization is the process of making the bacteria tastier. See, please don't write that it is the process of making the bacteria tastier in your exams. Please try to write the definition that is given in your books only, okay? This is just for the sake of your understanding. So, opsonization, basically what it do? It coats the pathogens. It makes the bacteria tastier. This is the bacteria. Some of the taste makers are there. Makes the bacteria tastier, makes the bacteria tastier. Right. Next is there is the complement activation. There is something called as the classical pathway, which leads to the lysis of the pathogen, which leads to the lysis of the pathogen. Okay. Then what will happen? Then there is agglutination and precipitation. This means the clumping of antigen for easier clearance. Next is the natural killer cell will come into the action. The natural killer cells kills the antibody coated the target cells. They basically kills the antibody, right? Next, let's learn about the T dependent and T independent. Okay, what are the T dependent immunity and T cells independent immunity? So the helper T cells are involved in T dependent. T independent, they are not involved. Antibody produced, the antibody which is produced by T dependent is IgG, and in the T independent is IgM. Memory cell formation is there in the T dependent, but it is rare in the independent one. The examples are in the T dependent is the protein, and here in the T independent is the polysaccharides. Okay, so see, it may not be asked, but if it is asked now, it will be asked as a 2-mark question only, not more than that, because it's not that important. Okay. Next, coming to the clinical relevance, because we are clinicians. So the clinical relevance is, first one is the immunodeficiency. So the defective B cells, if it is there is defect in the B cells, then there is recurrent bacterial infection. There is recurrent bacterial infection is there, and in the genetic section, I have told you, in the genetics, I have told you a disease in which boys are affected, and it is X-linked recessive. What is that disease called as? I'll give you the name also. It is something called as the Brutons. What Brutons now? That you have to tell me. Next is the vaccines. Vaccines basically stimulate the humoral immunity. Third one are the allergies and the hypersensitivity, that are the IgE mediated type 1 hypersensitivity. And once we are done with the humoral and the cellular immunity, next we will study about the comparison between the humoral and the cellular immunity. After that, we will be jumping to the types of hypersensitivity. There we will learn into the details. Fourth clinical relevance is the autoimmune disease. They are the autoantibodies targets the self-antigens. The autoantibodies basically targets the self-antigen. Here is a summary table which I have given you, and these summary tables are obviously very important. Here I have given you the mechanism. First one, there is antigen. Then the B cell recognizes the antigen. Clonal expansion is there. The plasma cells get activated. There is antibody secretions, and the pathogens basically do the process of neutralization. So this was all about the humoral immunity. Now it's time to solve some MCQ. First one. The first one is the main effector cells in the humoral immunity is. The main effector cells in the humoral immunity is it the CD8 T lymphocytes, B lymphocytes, NK cells, or the macrophages? What are they? So they are basically the T lymphocytes. In the cellular immunity it was T lymphocytes, right? So in the humoral immunity it is B lymphocytes. Next, which of the following antibody is produced during the primary immune response? During the primary immune response, which one is there? Is it the IgA? IgA, I don't think so. IgG? No. IgE? E is for allergic. G is for goes outside the placenta. I think it's M. It's IgM. Okay. Next you have is which cytokine is most important for the B cells and the proliferation and differentiation? So it is the interleukin 4. And we will also learn about the interleukins, because you know what interleukin 1 is? It is for fever. It's for fever. Rest, we will learn about the interleukins separately also, and I will give you certain mnemonics that you need to remember, right? So this was all about the humoral immunity. Now I guess it's time for us to read about the difference between the humoral immunity and the cellular immunity, right?